Hello, everyone. I'm Dr. Maisami, and call me Ella, please. I am a trained GP who is now full-time working as a translational neuroscientist and lecturer in neuroscience at Keele University. I am representative of Keele at the Midlands Immunology Network and joined the team, fascinating and fantastic group around a year and a half ago. My research interest is in neuroinflammation, systemic inflammatory response and central inflammatory response following neurological injuries and neurological insults. And particularly in COVID, very much fascinated by how immune response can modulate our nervous system and causes long-term sequelae in peripheral and central nervous system. Today, I am introducing two fantastic speakers. I hope I do them justice as I introduce them to you. First speaker is Professor Danny Altman. Professor Altman is Professor of Immunology in the Faculty of Medicine, Department of Immunology and Inflammation at Imperial College London. He is a head of the Hammersmith Hospital campus and expert in HLA system, antigen discovery, T-cell, and natural killer cells. He has a special interest in severe bacterial infection as well as viruses, including Zika. And when it comes to autoimmune disorders, he is looking into microbiota, and we are talking about omics now, and some of the autoimmune disorders such as rheumatoid arthritis. He also has an eye on cancer immunotherapy, where he investigates the autoimmune immunotoxicity in these patients. When it comes to COVID, he has, again, long-standing research in this field, and one of his very recent publications in American Association for the Advancement of Science, he and his peers investigated the role of vaccines in individuals, in actual boosting the B-cell and T-cell response in individuals who had prior infection to COVID. So I was very much impressed and fascinated by this work. And the second speaker that we have, I have the pleasure to introduce him to you, is Professor Paul Moss. Professor Moss is a professor of hematology within the Institute of Immunology and Immunotherapy at the University of Birmingham. He's also deputy head of the College of Medical and Dental Sciences. However, we might know him because of his national and international roles in translational human immunology and being a senior NIHR investigator. He is currently leading UK's Coronavirus Immunology Consortium, and this is they started this research about a year ago. So last summer, August, they received about £6.5 million fund from UKRI and NIHR, and he pulls together all the scientists, translational, clinical, together as a multi-center study to look into virus-specific immunity and how these responses could also relate to clinical outcomes in patients, particularly in different subgroups of patients. So now we are talking about stratified medicine and hopefully at some point personalized and precision medicine. He and his team also look into maintenance of long-term immunity and vaccine production and new therapies as well. He has several publications, of course, in Nature Blood and European Journal of Immunology, and his work has been featured in the Financial Times, BBC News, and Economist. One of his recent research publication in COVID was the uptake study that I found it fascinating. Just to briefly tell you, Paul and his peers looked into the study design trials that we do around vaccinations and looked into why we have less diversity when it comes to ethnicity, age, and comorbidities when we design these vaccination trials, and how we can improve our future trials. So without further ado, let me welcome, and hopefully you join me to welcome, Dr. Professor Danny Altman. His title of speech is a research program to look at the immunology of long COVID, inform diagnostics and therapeutics. Welcome, Danny.
Hello. Right, I'm going to try and share my PowerPoint. So I'm hoping that's okay now. Yeah, so I'm going to take a few minutes to try and introduce long COVID and convince you that immunologists hopefully have something to say about long COVID. And as you'll all be aware, we need to say something about COVID, long COVID, because it's a large and growing problem. So, you know, it's been a busy year and a half for all immunologists, hasn't it? And as it should be. So, right back at the start of the pandemic, if people can remember back that far, I think we all felt as immunologists that we had to stick our head above the parapet and say something and do something. And one of the first things that we did was to write this piece in the Lancet. So when I say we, it was myself with Rosemary Boyton and Danny Dueck. And without being all kind of argumentative about it, we felt that policymakers all over the world had suddenly become immunologists and virologists and epidemiologists and were experts on terms like antibody and PCR and herd immunity. And yet it felt important to slow them down a little bit and really explain in real life terms what that would mean for management of the pandemic and how one might apply those terms and what the limitations of those terms were. And it's a discussion that I think has stood the test of time, as in we're still debating those things and trying to get people to understand what we're talking about and why it matters. And then we started to pull together, both in our lab and looking across at other labs, you know, the overview of, you know, a whole new human immunology that we had to decode, which, as we were talking about in the last session, has really, you know, I think all immunologists should pat ourselves on the back that all that toolkit that we spent the last many decades developing wasn't just for fun. When we really needed it, it did come right, and it has served us well, and it served us really fast. I mean, you know, that tsunami of data that came along has, you know, has been for a purpose and has served us really well. So we did that, you know, sometimes I look at questions from earlier on in the pandemic, and it seems like a lifetime ago, and then I suddenly realized it was only 6 months ago or something. So our lab, because I'll mention it in a minute, we do both work in the UK, but also work in several other countries, including big cohort studies in Brazil. So we were one of the first labs to publish on the point that reinfection is not uncommon, and as we'd expect, reinfection is kind of correlated with being at the you know, the lower end of the distribution for protective immunity from your first infection. So we got heavily involved in trying to work out what were the you know, correlates and predictors and prevalence of reinfection in this disease. And then this is one of those, sorry, let's go back. This is one of those bits of the immunology of COVID that we all take for granted now. The idea that if you've had prior infection and you then go on to get vaccinated, you've got this absolutely enormous increment in your response. And it's one of those stories that was so obvious to spot in the data that we came up with it at the same time as Florian Kramer's lab in the States and John Werry's lab in the States, and probably one or two others. And so we all claim it and bicker over it. But I think this paper came out first. But it you know, it had big ramifications, and some countries actually switched to a one-dose vaccine regimen for people who had documented prior infection. But it's, you know, it's become a sort of real life piece of immunology that that's you know, that's interesting and has implications. And we, like lots of other people on this call, did lots of our work in in a large UK healthcare worker consortium. So this is a different one. This is the one that goes by the name the COVID Consortium, and it's the healthcare worker cohort that pulls together people at particularly Bart's Hospital, Royal Free Hospital, and UCH. And we've published many papers on different parameters in those individuals. And I won't go into all the details of it because the focus for the next few minutes is meant to be long COVID. But the idea of trying to map the detailed picture of immunity in a very large cohort across their kind of diverse clinical history, from uninfected to kind of should have been infected but weren't, to asymptomatic, to mild, to less mild, across mapping T-cell epitopes and neutralizing antibodies, and the concordance or discordance between them. And by the time we'd finished doing the first few rounds of that, and we thought it was all done and dusted, variant sequences came along that we had to incorporate into the story. And this is a version of that I think is still standing the test of time up to a point. But like many of the so-called COVID experts, I was kind of shooting my mouth off, saying that actually I thought that our kind of protective headroom from the vaccines was so magnificent, and we were seeing such enormous neutralizing titers that most of the world of in inverted commas normal people who'd made a good immune response to those vaccines would be safe against most variants for a good few years to come. But we did leave a kind of gap in the paper to talk about other variants that would stress test us and challenge us beyond that. And the ink was barely dry on the paper when Delta came along, and has made it look not completely foolish, but certainly has been challenging. And we've gone on to extend our analysis, like other people, into the relationship between homotypic and heterotypic immunity, as we have now we have a kind of complex world, don't we? We started out working out the rules for people who were infected by that kind of prototypic Wuhan HU1 sequence, challenged by it in the community, and protected by a spike sequence that was that original isolate. And now, if you think about it, we're inhabiting a world where anything's possible. You could have had an acute infection with one, a vaccination with the same thing, and then challenged by alpha, beta, gamma, or delta, or mu, or you could have a prior infection with any of those and then get your vaccine. So we have many different shapes and sizes and flavors of COVID immunity on the planet now. But what I'm meant to be focusing on, so I'll focus down and try and concentrate, is long COVID and where that fits in. So we were very keen on long COVID and very sympathetic to long COVID. I think anybody who works in this area or has been on planet Earth in the last 18 months very rapidly met people who said, hang on, there's something weird going on here. I had my acute infection, and 1 or 2 or 3 months have gone by, and I used to run marathons, and now I can't, or I can't walk upstairs anymore. And it was clear that this wasn't, this really wasn't the common cold. It was something different. And this is artwork from somebody we're actually working with on our project in our kind of long COVID communications. She's an artist called Monique Jackson, and she does this brilliant Instagram where she kind of posts her long COVID experience in real time. And this is that thing about I think I'm ill, I've got long COVID. You must try and relax, which I guess would make you even less well. So we all know about long COVID. In some ways, I think folks in the UK are often talked about as kind of exemplar. That we should be proud that we had a government that was fairly early on recognizing that long COVID was a thing and putting resource into it. And depending on your postcode and your GP, you can get referred to a long COVID clinic. And there's probably many people on this call who've been putting in the hours working in long COVID clinics. But I think it's obvious to all of us that it's challenging, isn't it? We all know the stuff about, how will you get referred there? And when you get referred there, which clinical specialties will staff them? In my experience, a lot of them are kind of led out of respiratory consultants, but what specialties do you need in there, and where will all these extra staff come from, and what expertise do they need, and what equipment do they need in their clinic, and what tests are they going to do in their clinic, and how do they deal with the fact that these are really, really complex patients, aren't they? So people will be very familiar with that. One of the very big patient surveys that was published in one of the Lancet journals in Eachley Med by Hannah Davis and others, and lists different permutations and combinations of 200 common symptoms, often relapsing and remitting. So somewhere either in that clinic or easily referred on from that clinic, you need your rheumatologist and your cardiologist and your endocrinologist and your respiratory team and your neurology team and your psychiatric team and things. It's a complex operation. So once you've done all this, what are you going to do for people? What therapeutics are you going to offer them to actually improve their lives and get them back to where they want to be? And how many years are the clinics going to run for? We've got all bets are off. We've got no predictions of is this a 1-year thing or a 2-year thing or a 5-year thing or a lifelong thing? We desperately need research to get some kind of predictors of the, if you like, the kind of symptom decay timeline of how long this is going to go on for. So Rosemary and I put together a kind of long COVID manifesto quite early on in the BMJ at a time when, I don't know, I felt, I know we need epidemiology and case counting, otherwise we don't know how much resource to put behind it. But I think I was feeling cross that all we had was epidemiology and case counting and nothing else. So one of the things we said in this piece was that we did need to move beyond observation to intervention, and that meant working out some mechanism and what on earth was going on. So I know different people now have their favorite hypotheses. The ones that I put forward at the time were the ones that seemed to me the obvious. So this is a kind of and/or kind of list. They're not mutually exclusive. The idea, the kind of thing that comes up on the imaging studies, that if you've had a fairly cytopathic virus in a load of ACE2 positive cells all around the body, lung, liver, kidney, heart, blood vessels, what have you, you're going to see some scarring and some fibrosis and some tissue damage. It'll take a while to recover. And one can certainly find evidence for that in the imaging. Or the kind of the kind of herpes virus, CMV, EBV kind of idea that if you've had a virus doing a lot of stuff in your immune system, it's a bit like kind of chucking a hand grenade into the immune subsets. It's going to look disruptive for quite a while to come. The idea that we're quite keen on that, again, growing support for the idea that it could be a reservoir of persistent virus. For example, all that data on either staining or PCR or culture of virus out of the gut and out of stool samples and things, or the idea that I'll focus on for a few slides, that actually it's a virus that can induce, bona fide de novo autoimmunity, and lots of growing evidence for that. One of the reasons we were so keen on that and had a kind of background in that is that we have 2 big consortium cohort studies going in Brazil together with a sort of co-PI there, this chap at the bottom, Andre Seguera. And so we have one cohort study on chikungunya called REPLIC and one on COVID called REPRA-COVID. And for those who don't know and love chikungunya, it's a bit, it's obviously it's one of the the arboviruses that you get in hot countries where Aedes aegypti mosquitoes nip you, and if they haven't given you Zika or dengue or yellow fever, they might have given you chikungunya, which is the reason it's kind of devastating is that even once you've had your your rash and your fever and your malaise and recovered from your acute infection, perhaps 30 or 40% of people have a kind of autoimmune arthralgia that goes on for years and really destroys their lives and their employment and their income and the health service. So, you can see where I'm headed with this, that it had a kind of resonance to the kind of things I was hearing about long COVID. So that, yeah, that's just, you know, to set the scene, the kind of stuff that we talk about in chikungunya research. So we were one of the labs that was funded by NIHR in their call on studies to diagnose and treat long COVID. So I just wanted to talk about the kind of thing that we're trying to do there. So so bear with me on this. It's fun. So this is a little pilot experiment that kind of positioned us for where we needed to be for this. So amongst all the projects we were doing before kind of life collapsed in you know, January, February 2020, we had a big, and still have a big collaborative program with David Pinato at Hammersmith Hospital and Imperial College, and he heads the oncology immune checkpoint register there. So you know, as you know, immune checkpoint blockade is an absolutely brilliant way of treating many different cancers, but in many people is kind of correlated with quite a high cost in terms of induction of autoimmunity and you know, immune toxicity. So this is a little pilot experiment that we did before starting to work with him, and it goes like this. You take serum from mice, that's the top bit. Oh, sorry, let's come back. Or you take serum from mice that you've made systemically autoimmune. So these are mice that on the one hand are FOXP3 inducible deletion mice to get rid of all Tregs, and on the other hand are gamma interferon luciferase mice to report activation of the immune response. And what you see is all of the obvious. You deplete all of your Tregs, and as has been known for many years, the mice become autoimmune in every organ, and you can see that lighting up in real time in your bioluminescence imaging. And you can also see it like this in this kind of starry night, because what this is using a whole proteome array to see which autoantibodies light up in terms of binding to bits of the human genome. In this case, used as a sort of proxy for the mouse genome in terms of you know, cross-species, cross-reactivity. So we regarded that when we set out on our autoimmunity work as a way of kind of looking at whole genome autoimmunity. So we went on to do that in long COVID. And to cut a long story short, what we've been up to in recent months is making friends with everybody we can possibly make friends with who wants to join us to look at long COVID. And, you know, always happy to make more friends if anybody wants to team up with us. So we've worked with, you know, post-COVID, as in, you know, the post-hospitalized COVID cohort. I said that we've done a ton of work in the COVID consortium in London. And, you know, I think it goes without saying that if you've got a cohort of many hundreds of people, and within that, you've got several hundred people who've had COVID, if we're saying that somewhere between 10 and 20% of those, no matter how mildly they were affected, have persistent symptoms, you know, that's a resource for looking at long COVID, not just at COVID. So, you know, to do it. We're very keen on CoverScan, which is the kind of spin out of Oxford University that's correlating long COVID with, you know, with MRI, whole body imaging. We've got together with lots of the other NIHR awards. We've got our, you know, cross Brazil 9-center study, which is interesting because obviously they're looking at gamma, not at alpha or delta like we tend to be. We've teamed up with ISARIC, which you'll know is you know, one of the largest cohort studies in the world. Our main award was actually to look at all the others that don't get looked at in these studies, which is all of the people who never got hospitalized, maybe never even saw a GP, maybe never even had a PCR test, who know they've got long COVID and just desperate for somebody to talk to analyze their disease. So we're reaching out to them through the support groups and long COVID clinics and GP surgeries and adverts. We're also looking at the Stimulate cohort and at the pediatric cohort through the Evelina, and doing those kind of starry night assays on the whole proteome arrays. And obviously not all autoimmunity is just autoantibodies. Got lots of T-cell aficionados on this call. So we're also using the autoantigens that we get out of the proteome arrays to lead us to T-cell assays. So it ends up, you know, like all of these arrays, you can present it any way you like, but it ends up looking like this on heat maps and things, where you can decode them to look at which parts of the proteome become targets for autoantibodies. And sure enough, that, you know, there are rather specific signals that you can pick up in long COVID that you don't pick up in your kind of Matt Hancock type of person who, you know, had COVID and then a week later was jumping around all perky saying, I feel recovered. So there is a differential signal, and you can map that to particular antigens. And we're trying to do more and more of that at the moment to, you know, to pin down what the signal looks like, married up to, in the first instance, to the MRI changes on cover scan. So that was a sort of whistle-stop tour. These are the people who've done all of that work, and those are some of the collaborators who've been doing it in lots of places. So there I'll stop sharing, and I guess I'll take questions after Paul's talk.
Thank you so very much, Danny. That was fascinating. Yes, indeed, we are going to allow or open up the QA after Paul has completed his talk. So please join me to welcome Professor Paul Moss with the title of his presentation, Determinants of Immune Response to SARS-CoV-2 Vaccines Across the Life Course. Welcome, Paul. You're muted.
Yeah, thanks very much. It's great to be with you. Can you hear me okay? I hope so.
Yes, indeed. Thank you.
Super.
Feel free to share presentation or whenever you're ready.
Yes, I will. Let me just get this sorted. Nice talk from Danny. I don't do a lot of work on long COVID myself, but I want to talk about vaccines today because I believe that they're going to be very important in the management of long COVID. So let me share my screen and then we'll get going. So I want to talk today, I think it's 15 minutes, about immune correlates of vaccine responses in long COVID. So the outline here, I'll talk about vaccination in immune competent people. I want to talk about the influence of aging and vaccine choice, dosing interval, and then we'll talk about immune suppression. And I'm very grateful that you've invited me today, and it's great to have the Midlands talking together with so many colleagues and friends on the line. I've just seen and the participants, and I recognize a lot of them. But in relation to this section on long COVID, is vaccination important? Well, yes, it may well be. And some reports suggest that vaccination can help symptoms of established long COVID if you've got long COVID after a natural infection. The question here is, does it help to eradicate residual virus or residual protein? I mean, that's quite an interesting concept. I'll address that in a minute, because this is supposed to be an acute infection, right? Like influenza, you get rid of it. So that's interesting. This study, which Nature reported on, it wasn't published in Nature, but they looked at 800 people after the vaccine, and about 57% said it improved, whereas 19% said they were worse. So I don't, it does suggest that data. And this came from the Zoe app. About 1 million people or more around the country are following this app. And so if you look at people who got breakthrough infection after vaccination, then they seem to have residual symptoms of around 5% at a month compared to previous data on 11%, suggesting that if you've got a vaccine on board, you may get less long COVID. So that's also generated a lot of interest. And so this is the sort of interesting data that was published certainly in preprint last year. And this is the suggestion that viral RNA and protein may actually persist in your body. So all of those of us on the line who've been infected with naturally, I don't know, what is it, a third of us maybe? It's 50% of children. It's extraordinary. A lot of us have had natural viral infection, and we may still have some viral protein that's hopefully not replicating virus in the GI tract. They looked at 4 months after infection, and they detected protein and mRNA in a few donors. So yeah, raises some potential for viral persistence. And with that on board, let's talk about vaccine responses, which my group have spent quite a lot of time working on in the last year. And I'm going to start with older people. And what do I mean by that? People over the age of about 80 years, because we've been interested in this phenomenon of immune senescence for quite a number of years, actually. And there's two groups of older people that we're studying. People who live in the community, in their homes, maybe with carers or totally independent, and those who live in care homes. I remember being asked at university many, many years ago, what percentage of people, older people over the age of 80 or whatever, live in care homes? And of course, when you're 20, you think, oh, most people surely in care homes. It's actually a very small percentage. It's way less than 10%. Most older people are independent and living at home. And so we've studied around 300 people in the community, and goodness, with Vivaldi, I think we've got over 10,000 samples from the UK care home study. And I'll talk more about that later. So we've got two interesting cohorts here. Let's start with community dwelling people. Now, the vaccine around the world, America, Pfizer, Europe, is this regime largely. Pfizer, BioNTech, 3 weeks apart. That's what it's licensed for. And when we studied this in a group of people, this was Helen Parry, Rachel Bruton, collecting people in December last year, they started to recruit. And then we looked at their antibody responses after 2 vaccines. And this is people over the age of 80 up to 100 years. This is our antibody response. Stunningly good. Everybody responded. One little slightly lower response. Everybody. Incredible. We couldn't believe the data we were getting back from Porton Down. So this is a tremendous vaccine. And of course, you're not surprised by that because of the protection it gives you. Protects you against death by around 10 or 11 times. I mean, just extraordinarily good. So super antibody responses in older people with this Pfizer. So can we do better? Well, maybe we can. We'll talk in a minute. The other thing that came out, and lots of groups have seen this, is that if you've had a previous infection and survived it, of course, looking at survivors here, then you get better antibody and cellular responses. These people in blue were the ones who'd been vaccinated having had a previous infection. And you can see they make super antibodies. Even after 2 vaccines, they are better. There you see them standing out right at the top of the antibody responses. And so this natural infection, if you're going to get a natural infection, as Danny says, it'll be Delta now, but it's going to give you, assuming you get through it, obviously vast majority of people do now, going to give you good immunity. Now, that is the international standard, 3 weeks apart, BioNTech Pfizer vaccine. But as you will know, in January last year, just as we'd started this, the government and JCVI said, no, we haven't got enough vaccine, and we also can't deliver it quick enough. So what we're going to do is go to an extended interval. So you're going to have to wait 10 to 12 weeks to have your second dose. Now, I must say this was actually very surprising. I was surprised myself, but they were right. They were right. They were right because modeling shows that if you've got limited ability to get vaccines out into the community, and all of us have been to a vaccine centre, haven't we? There's only so many people you can get through in a day. And you may as well get 1 dose in. That protects you about 80% against death with 1 vaccine. 2 might give you 90% or more, but get 1 dose in. It was right. And the other thing why it may be right is it may be better for your immune system. Yes, even the Pfizer vaccine. So this is our data. This is the international standard. Get it in 3 weeks. Measure your antibodies. They're strong. I've told you they're strong already. But if you leave that Pfizer for 12 weeks and then measure the antibodies, wow, they are way, way better than after a 3-week interval. So if you leave the Pfizer for 12 weeks, you get much stronger antibody responses. This is the cellular response, peaking in blue after the 2 doses, and then peaking slowly after the second dose. You can see that there isn't any difference there. And to be honest, you know, that you could say that this data might inform international vaccine strategy. Maybe BioNTech, Pfizer should be going for this. We've just bled this cohort now at 6 months. And I think it'll be fascinating to see where this is at 6 months and where this is at 6 months. Because if this is still higher then maybe BioNTech do need to change, and it should explain why UK is possibly getting better protection with these vaccines than places like Israel. So if you want to split hairs about these two vaccines, both of which are great, the Pfizer or the AstraZeneca, then you can, in our hands, we see this. Pfizer is better at antibodies, but old AZ, the Oxford AZ, gives you stronger cellular responses. Here's the antibody responses. You can see after one vaccine, they're the same, no difference. But after two, you see that the Pfizer is coming up. It's boosting better for antibodies. Do you think antibodies are the thing that protects you against death? Who knows? We don't have that correlate. If you look at cellular responses, and this got AZ very excited a few months ago, you will find that after 2 doses, 1 or 2, AZ giving you better T cells. So my colleagues on the line, Heather and Lawrence and all those colleagues, those T cells are higher after AZ, and we're now again doing this at 6 months. We think we see the same thing. Now, we're also studying these people who are more frail in care homes. This is with Vivaldi. Laura Shellcross, running this national care home study, is running my group by Gokhan. We've got many people on the line I can see from Gokhan's group, Tara, Alishka and Megan and so forth, have been working hard and taking in a lot of samples. And essentially, if you've not had previous infection, older people have weaker vaccine responses. So here we are seeing some immune suppression in these frail older people. And of course, this is one reason why they need to get a booster this winter, and we'll be measuring that response. And that was antibodies, and the cellular responses are also lower. Ignore the red ones, they're previously infected, but in the blue ones, young, old, you can see the T cells dropping down in these older people. And Megan, who I think is listening, is now looking at this using very detailed flow cytometry. And so here you can see that if you've had a previous natural infection, you're getting T cells against spike and other proteins. Whereas if you're relying on your vaccine in these elderly people over the age of 80, you're only getting these spike T cells. And she's trying to understand the difference between the quality of these T cells after natural and vaccine-induced immunity. So that's extreme of ages. What about children? Our children make super immune responses to natural infection. You know, one of the incredible things about this infection is the risk of aging. Every sort of 8 years, you double your risk of dying from this infection. And Alex, who I think is also listening, did this work with children. And what we're seeing is very strong, sustained antibody and cellular responses after natural infection. In a way, that's no surprise. Your best time for your immune system to protect you against death after infection is around between the ages of around 5 to around 15. As soon as puberty kicks in, your relative protection starts to drop off. Immune senescence doesn't start at 60, it starts at puberty. And we published a paper with Judith Glynn on that a couple of years ago. And so we think there's a lot of cross-reactivity in children. And Alex is now studying vaccine responses in these children. But of course, there's a lot of interest and debate about vaccines, but we do have some from primary school children. But I just want to finish really on comparing to immune suppressed people. There are around 500,000 people who are immune suppressed. So I don't know, if we got 50 on the line, it's 10,000 people for every one of us. It's a lot of people. And these are people with cancer diagnosis or on renal dialysis or had a solid organ transplant like a kidney or something like that. And their antibody responses are poor. And we have studied one group of patients, which is these people with chronic lymphocytic leukemia, where they've got too many of these, their B cells actually in their blood, and it makes them, very immune suppressed. And we've studied 500 people, both around Birmingham and across the country, using dried blood spots, where you can send a little sample of blood through the post. And this was led by these colleagues, Helen Parry, Rachel Bruton, Graham McElroy. Helen particularly has been leading this. funded by NCS and UKRI, and charities have been super as well. And this is the summary. That's hopefully us on the line, healthy donors, 100% antibody response after 2 vaccines. And this is this group, 67%, a third are not making antibody responses, no antibodies. And even if you look at those who are, their antibody responses are low. What are the lessons? There are some interesting things. As immunologists, I think you might be interested to see that the responses to BioNTech, mRNA, and AstraZeneca are the same. AZ is not inferior here, nor is it indeed superior. And here we've looked at live virus neutralization. These are you and me, healthy donors. This is how we neutralize the original Wuhan virus after vaccination. And this is the Delta. Delta does evade the immune system to some extent, not strongly, but significantly for sure. This is CLL patients. You see they drop down against the original Wuhan, and look at Delta, which is the only virus in town now. The antibody neutralization in these patients is pretty diabolical. This is across their disease course, by the way. So this is when these patients with leukemia are untreated. Many of them are. Their antibodies aren't too bad, but as soon as you start to give them treatment, the antibody response to vaccination drops down. And once they've finished treatment and they're, recovered or not cured, but long-term remission, it starts to improve. And so the state of disease is a determinant. So what do we do? Well, we've been debating, and studying this third booster vaccine, and we've put many of these patients, 50 have been vaccinated in Birmingham now, into this Octave duo, which actually I think is on the next slide. And it's this run from Ian McInnes, Pam Kearns, and they're comparing a third dose of Pfizer or Moderna or Novavax. And you'll know Pfizer, many of you have had Pfizer. Moderna is very similar, perhaps a little stronger. And Novavax is a protein vaccine. And so we are assessing whether a third dose and which third dose is better. And the other approach, which is very exciting, is if people can't make an antibody, you just inject a therapeutic monoclonal antibody, which can last for many, many months. Old-fashioned approach, passive immunity, injecting antibodies. So I'm just going to finish up there and say, current vaccines are excellent. I mean, we're immunologists today. I suppose it doesn't really matter about the immunology. The clinical is excellent as well, but the immunogenicity is. But they differ. They differ in subtle ways. There is some suggestion, you've probably heard this, that the mRNA Pfizers give you strong immunity, but wane more quickly. And the AZ is more like the tortoise in the race and may actually sustain immunity for longer. The dosage interval may be critical. The 3 weeks was picked really much out of nowhere because this vaccine came through so quickly. It may be that should be extended. They don't completely overcome immune senescence in older people, but they're pretty good. And of course, patients with immune senescence have suboptimal responses, and we need to overcome that. What we're doing now, well, as I say, it's longevity now becomes the question. In many ways, you could say, well, everyone's going to have a third dose anyway, everyone over 50 indeed, quite remarkable, but we will need to study that. And we're assessing things like neutralization and cellular function, of course, policy really, and as many people have said, it's great to see immunology guiding policy. So I just want to finish with a few acknowledgements in terms of the elderly cohort and the CLL, Helen, Rachel, Jianmin, Graham, but many, many others in the lab. And the Vivaldi has been led from our team by Gokhan, but as I say, I've name-checked quite a few people. And indeed, Alex Dowley works on the children. And Laura is our great collaborator with Vivaldi. And the funding has come from UKCIC and National Core Studies. So I think that I'll stop there and take some questions with Danny.
Thank you very much, Paul. That was fascinating. So let's see how many hands are up there or any questions. So I don't see any questions from the audience. Let me take a start with some okay, the first question goes to Danny. You very nicely showed the importance of prediction and predictive biomarkers. Where do you see the a computational, predictive modeling, or have you already utilized this approach? And any comments? That would be lovely to hear it from you.
You mean predictive for long COVID?
Yes, please.
Yeah. So it's early days. The answer is that people are worried about long COVID. They're terrified of long COVID. So predictive markers and working out who's going to get it and what you could do about it before it's too late would be really, really important. So the sort of predictive analysis is really early days. It's like the sort of meta-analysis that comes out of those big data sets, and we and everybody else is working as fast as we can to try and make that a little bit more sophisticated, and put some more meat on those bones.
Excellent. That's fascinating. Yeah, I look forward to see those publications. And next question from Paul. That was fascinating talk again. And let's talk about the stratifications of these groups. Do you think, you mentioned that the, when you compared the vaccine AstraZeneca versus Pfizer, you could segregate patients who are below 65 and above. So we stratified them by age. Can you comment on various ethnic groups? Have you looked into it?
Yeah, that's an interesting, incredibly important and topical question, isn't it? And it's one that we wanted to address. We didn't really address it quick enough, quite frankly. So in our cohort, we can't address that. Through the National Cost Studies work, and Lucy's involved in this, we've got cohorts established in Leicester. Of course, Andrea that is doing this, right? So UK REACH program. So that is much more powered to address that question rather than us.
Thank you. Yeah, it's important to consider for future, perhaps.
Yeah.
Next question from no, not audience. Lucy, please. Yes.
Danny, can I ask you a question? So I think the challenge that we've got with long COVID is the fact that it's this really massive umbrella terminology that I'm sure is going to encompass many, many different sort of reasons for getting long COVID and even symptoms or clinical outcomes of getting long COVID. So I guess my point is, how do you think we're going to manage that diversity with this sort of really big umbrella terminology that we're using?
It's like so much for medicine, isn't it? That every meeting I go to, there's a kind of divide between the stratifiers and the lumpers, and and obviously a lot of modern medicine is nearer to the stratifiers and the personalized medicine. So I suppose the logic that we follow is, sure, on the one hand, why should somebody who's suffering symptoms because they've got lung fibrosis look the same as somebody who's suffering brain fog because maybe they've got some autoantibodies against, a neuropeptide or something. So I suppose we're trying to kind of walk an agnostic line straight down the middle and look as comprehensively as we can and agnostically as we can in terms of the whole proteome arrays and the autoimmunity and the immunology, accepting that might not be the answer to everything, but we're also hedging our bets by doing some stratification and leaning quite heavily on the sort of stratified end organ damage that one sees in that cover scan MRI cohort. So trying to kind of have our cake and eat it, really.
Andrea,
I think that's the biggest challenge. Can I just ask one more question as well of Paul?
Yes, please. Okay? Andrea, please wait. Yes.
So Paul, I don't know if I've seen any data, but what do we know? You said about Pfizer and AZ vaccines either driving a really good cell-mediated response or driving a good antibody response. Do we know at this stage how that relates to hospitalizations in those individuals, those few individuals that have been hospitalized after vaccination?
Yeah, it's an interesting question. I mean, I drew that extreme apart, and we do see slight differences, but of course both vaccines do generate strong antibody and decent cellular immunity. I mean, certainly the Pfizer gives good cellular immunity. It may be that just certainly in these older people, we're seeing a little tweak more with the AZ. That correlative protection that you asked for is the key thing we want. It sounds like a dream, doesn't it? That, if you measure your spike antibody and it's 100, you're 95% protected. Will we ever get, or 90, more than that? Because will we ever get to that? Well, I mean, it's not inconceivable. And We talk a lot about concepts like vaccine passports. Vaccine passports have been around for decades. You know, people like myself and Lawrence have been to Africa. You need a yellow fever vaccine, right? You have to do it. And we use antibody levels to guide what we think is protection against hepatitis or pneumococcus and things. So it's entirely possible that we could get to that, but we don't have it yet. There's a lot more interest in cellular immunity. The only infection, I think, where people used to think about T cells was TB, wasn't it? And that's where people have been measuring responses, as Andrea said, but maybe whether we'll be doing it in COVID, I don't know. It's an open question. I hope, as a T cell biologist, it would be nice to think so.
Thank you, Lucy. Andrea.
Yeah, so just to follow on what Paul said, I think if we don't have one after all of the work we've been doing, and I think we'll just have it a lot quicker than we would have expected. And I think coming over to Danny, actually, this is maybe a provocative question, but I wonder if psychological approaches with long COVID, are we able to investigate, do you think, if not neuroimmunology, but psychoimmunology, in terms of if we actually know that there's a physical problem with a particular organ, is what you're getting with your with the scanning, so you can sort of tap into that, and that's a great confidence builder for the patient. But if you can't find a proximal reason for their illness, what can we do about that? I think to me it's really interesting that and that drawing they had from the patient, and so we are collecting qualitative interview data in UK Reach, and I wondered if that's part of what you're doing with the how much that will be integrated into your sort of more hard data immunology.
Well, I mean, you know, there's plenty of neurologists and psychologists and psychiatrists looking at long COVID and sitting in long COVID clinics and being referred patients. But, you know, I think immunologists have as much right to be delving in people's brains as anywhere else in the body. You know, so I'm not, if people's symptoms are more at the kind of sort of, you know, CNS end to do with, you know, anxiety or hallucinations or memory problems or anything, I'm just as happy to look at the immunology of their CSF and their CNS imaging and things as I am to look anywhere else.
But I'm also thinking of the effect of stress on immunological outcomes,
sure.
So it's a more generalized impact. And I think that applies to our TB studies as well. You know, just simply understanding the patient's approach to their own disease and how that influences the expression of they have an immune response, but is it going to be an immunity, a protective response, if they're compromising it by corticosteroid levels associated with stress that's not that we can overcome by some intervention. Probably outside the discussion for this, but I just wanted to bring it up.
I just think if we're going to uncover that, we've got to be much more aspirational in our kind of multidisciplinary teams and holistic approaches. What else can I say?
Yeah, it's fascinating what you mentioned, Andrea, and Daniel, of course, followed with CSF. I inquired UKRI, and unfortunately, they are not collecting MRI images from brain or CSF. So that's something I wanted to do a year ago, but that should be done at regional hospitals or in a collaborative form with peers who are interested to work with translational or basic scientists like myself, and there are also clinician neurologists. That's fascinating. I have two questions from audience, if you don't mind me. Cole Edwards asked, I think that might refer to Paul because of the nature of question. You said you found vaccines seem to reduce long COVID effects in the vaccine cohort. Did you have any patients with long-term effects from previous coronavirus infections, such as existing anosmia? Did you find that the vaccine effect alter these symptoms? Or how much is your knowledge specific on anosmia on this?
Thank you. Yes, well, actually, that work I was quoting was not ours. So that's that reference to this Nature. It's not a Nature article; it's a commentary in Nature. So I can share that. That was just a study showing that at 800 people with long COVID symptoms who had vaccination, more people showed an improvement in symptoms than a deterioration. So I don't really know the details, and it wasn't my own work.
Okay.
So I probably can't comment further
That's
usefully on
Thank
that.
you for sharing your thoughts. Second question before we hand it to Lucy and the next speakers. Anne is asking about implications that SARS-CoV might have in the first trimester of pregnancy and the fetal development. Any of our speakers might be able to comment on that?
I don't think we've seen any particularly worrying or concerning data of any kind of cross-placental infection or anything, have we? I don't think we have.
We didn't cover much of comorbidities apart from immune suppressions, chronic or acute, but that could be a whole new webinar talking about vulnerable patients such as pregnant and all those with comorbidities, obesities, and all other autoimmune disease. Thank you so very much, both the speakers, Paul and Daniel. It was a delight having you. Going back to Lucy, and yeah, thanks again.